Good afternoon, everyone. I'd like to welcome you to the 2017 version of SU Research Day. Here at Salisbury University, we really value the contribution of our faculty and their scholarship, and we appreciate you being here. This is our 5th anniversary of SU Research Day, so we're really happy about that. Glad to see students in the audience. This is the first year we've reached out to students and you've showed up, and I'm sure none of you are getting any extra credit whatsoever for being here, and that's why you're taking notes even as I'm just doing the introduction, I'm sure. But we're glad you're here and we hope you get a lot out of it. Want to say thank you to our interim provost, Dr. Karen Olmstead, for being here. She's always a great advocate for research, for students, faculty, everyone on campus, so we really appreciate her efforts and time and energy, and thank her for being here. I'd like to thank everyone who is participating between 5 and 7. We're going to have a reception as well to talk about the importance of mentoring. So all these fine faculty that will be giving their talks are also dedicated to helping mentor students, and we really appreciate their efforts on behalf of our students as well. Next, we have from the Department of Biological Sciences, Dr. Jessica Clark. The title of her presentation is Earning Your Stripes, Undergraduate Research in a Zebrafish Lab.
I'm embarrassed to tell you how long it took me to come up with that title. Let me just put that out there. First of all, thank you all for coming out today and taking part in Research Day. I want to thank Dean Griffin and his amazing team for putting such a great day together. And I also want to thank Dean Scott for giving me this opportunity to represent Henson. It really is quite an honor. So I'm probably going to lighten things up a little bit, because what I do is I make baby zebrafish that fluoresce diabetic. And then I section them and I look at their nerves. I look to see what the hyperglycemia, which I'll talk about in a minute, is causing their peripheral nervous system to do, okay? So I know we have a pretty broad audience, so I'm going to talk about the nervous system. And you probably remember this from maybe Intro to Bio or Intro to Psych. But the nervous system is divided into a central component, which you can see in the red, it's the brain and the spinal cord. And then you can see in blue the peripheral component. And this is making up all of the nerves that connect our brain to the environment around us. And so if you remember the classical reflex arc that we've all learned about in our gen ed requirements, you can see that the information comes in through activation of the sensory nerve. That information goes into the spinal cord, and then it projects back out through the motor nerve. So these are the two components I'm going to talk most about today. I'm going to talk about the sensory neurons, and then I'm going to talk about the motor nerves. In terms of the motor nerves, what I spent the most amount of my postdoctoral work studying was a very important class of glia called paraneurial glia. So here are our axons right here, and this is what's carrying the signal, okay? And then we have Schwann cells that myelinate the axons to speed up the signal so that we don't lose any of that very important information. And then these are delicate structures, so they're encapsulated into fascicles by the paraneurial cells, so the green component right here. And hopefully I will impress upon you the importance of these cells. And the reason that they're so important is because this is what's making up the blood-nerve barrier. This is what's protecting the nerve, the axons, the myelin sheath. It's what's protecting these components from ionic flux and from environmental insult. My postdoctoral work looked at what happens in mice if we get rid of the paraneurium, and what we see is that the myelin sheath degrades. Okay, this is the sciatic nerve. And in green, you can see here the difference. The green is axons. So you can see the difference in the axons with the presence of the paraneurium and without the presence of the paraneurium. On an ultrastructural level, you can see the myelin sheath with a intact paraneurium, and then the myelin sheath starts to degrade when we lose that paraneurium. So the paraneurium is really important. So when I came here, I was interested in creating a model of peripheral neuropathy, looking to see if we disrupted the peripheral nerve and we made a clinical state of neuropathy. We can do this in a number of different ways, but the cheapest is to make a diabetic model, okay? And why diabetes? Well, I was at University of Virginia before I came here, so I was very clued into the idea of needing to have a clinically relevant model to get those NIH dollars. And so just to give you some background on diabetes and diabetic neuropathy, the World Health Organization is forecasting that by 2050, 1 in 5 people will have diabetes. We know as of right now, about 50% of people that do have diabetes suffer from diabetic peripheral neuropathy. So it's about a $14 billion cost just for the diabetic peripheral neuropathy. So it's a major issue. It's a major issue. Now I'm going to answer the other question, and that is, why zebrafish? Zebrafish are, in my opinion, one of the best animal models. I worked with rodents for years, developed terrible allergies. I haven't developed an allergy to zebrafish yet, but leave it to my immune system to do that. In terms of why we use zebrafish, there's a number of reasons. First, the genome is known, so that's incredibly useful. We can edit pretty easily. In terms of their growth cycle, we can have our fish, our zebrafish, will lay hundreds of eggs, so this is at the 2-cell stage. And by 5 days, we have a fully functional larva. Okay, so it's very rapid. And my fish specifically, because I'm interested in the peripheral nerve, I can actually fluorescently label their nerves, the different components of the nerve, and then see how these components come together. And in a disease state, how they actually fall apart. So that's what I'm going to show you today. The other reason is because zebrafish have this amazing ability to regenerate. And when we're talking about nerves, that's wonderful, right? But we're talking in zebrafish, they have the ability to regenerate their central nerves as well. So we're talking about things like MS, okay? They're able to regenerate cells in their heart. So you kind of get the idea. So when we're talking about degeneration of these peripheral nerves, I want you to imagine in the future in my lab that this is going to be a major component, trying to figure out how the zebrafish is so good at regenerating and how we can exploit that to let it translate to a clinical aspect. So here are my zebrafish. This is a picture that I took of the fish. So you can see we have 2 labels here. The green is labeling cells in the spinal cord. And then you can see in red, these cells are going to label the motor axons. I'm going to zoom you in a minute. But I just want to mention another reason we use zebrafish, and that is that the nerves, the components of the nerve are conserved between mammals and zebrafish. So we're actually a lot more like zebrafish than you would think. We can live image these fish. So we can put them directly on the microscope and we can look at their nerves directly while they're alive. Or we can section them and we can take very thin sections through their entire body. And then we can label, either through genetics or through antibody labeling, we can label the different components of the peripheral nerve to see how these components come together. And again, in a pathological state, we can see how these components fall apart as well. Okay, so hopefully this is going to work. This is some beautiful work that my postdoc advisor did when she was a postdoc. But this is another reason that this is a really important animal model to study for, especially for diabetes, because we have such a small amount of knowledge as to what's happening. We can take snapshots in humans, we can take biopsies, we can take snapshots in rodent models, but in zebrafish, the different components of the peripheral nerve in real time coming out. So in red, you have the motor axon, and then in green, you have the peroneural glia that I mentioned is the blood-nerve barrier. So we can see these things come together. Okay, so in terms of diabetic peripheral neuropathy, for those of you that don't know much about diabetes, I'm going to tell you that in our healthy body, our pancreas puts out a hormone called insulin. Insulin binds to receptors in our cells, and it puts these GLUT4 transporters into our plasma membrane. And that's what's going to get the glucose that we're taking in from our diet, for example, from our blood into our cells. Okay. Now, in a diabetic state, we have 2 different types of diabetes. We have type 1, where the pancreas stops making insulin, And we have type 2, where the receptors stop responding. And in both situations, what I want you to remember is simple, and that is that the glucose stays in the blood. It doesn't get into the cells that need it. And so what ends up happening, there are a lot of very complex chemical pathways that basically get junked up, for lack of a better term. And the bottom line is we see a lot of organ damage. We see a lot of cellular damage. In zebrafish, there's already established models in adults. And what it shows is that the adult models, we can recapitulate in zebrafish the pathologies that are associated in humans. So for example, you can see changes in the nephron in the kidney. You can see changes in the retina in the eye. And down here, we can see this delayed wound healing. But what's noticeably absent from the literature are the nerves. What's happening to the nerves? We know that 50% of patients that are presenting with diabetes have peripheral neuropathy, but yet there's this wide open question that still exists. So remember, we have two models, type 1 and type 2. Type 1, we're missing the cells that make the insulin. Type 2, we're missing the functional receptors. So lucky for me, there was actually already a model of type 1 diabetes in zebrafish. This comes from Mike Parsons' lab at Johns Hopkins, and he was kind enough to share with us some of his zebrafish. And so basically, we have the pancreas labeled in red here. And so in these fish, anytime the insulin promoter is driven, we're going to have not only insulin being made, but we're also going to have a fluorescent protein. Okay, so that's how we know the pancreatic beta cells are there. Now, what Michael Parson did was he also added this component here. This is a gene that encodes a nitroreductase, which is found in E. coli. And what's really cool about this component is that in all the cells that make insulin, we're going to have this nitroreductase. We can add metronidazole, a drug, into their water. It comes in transdermally. And what happens is it causes cytotoxicity in only the cells that express this gene, okay? So in other words, we can directly chemically ablate the cells that are making insulin. So I'm gonna start telling you about the research that we've collected over the past 2 years. And I'm gonna tell you, I wanna make sure that I'm giving credit where credit is due. And that is, my students come in and the first thing they do when they come into my lab is they write a grant. And so they have a very strong foundation in terms of literature review before they start their project. And then from there out, I am teaching them the technical aspects, but they're running everything. They are crossing the fish, they are collecting the eggs, they're running all the experiments, they're coming in on weekends to feed their fish, they're really doing it all. And then when we collect the data, when they collect the data, we go through it together, we analyze it together, and then they start writing it up. So this really is a testament to how hard they are working. Amanda Rocker, you'll notice a lot of these students are from the honors program. It's because they show up early and often, which is one of the things I love about our honors students. Amanda showed up on her first week of her freshman year because she knows she wants to get a PhD in neuroscience. So Amanda and Julia Howell just graduated last spring. She just started her first year at University of Kentucky doing a PhD in neuroscience as well. They asked a simple question: can we take these fish and can we prevent them from making insulin? And when we look at the blood glucose in the individual larvae, which is, you can imagine trying to get blood glucose out of a larvae, we see that the treated animals are in fact hyperglycemic. So this is proof of concept that this fish was capable of chemical ablation. So Julia, for her thesis work, she asked the first question, and that was, does hyperglycemia affect the perineurium? Is it affecting this blood-nerve barrier that we know is so very important? So just to acclimate you, on the side here, you can see the transgene. This is just its genetic background. Use my little key down here. In green, you can see the cells that make up the perineurium. In red, you can see the motor axons. Up here, this is the spinal cord in the zebrafish. These are the motor nerves that are coming out of the ventral aspect of the spinal cord. So in our control, we see exactly what we would expect, and that is these red projections of nerves. And for the majority of them, we see them nicely ensheathed by the perineurium. So you can tell that they're ensheathed because it has this nice yellow tone to it. Now this nerve is beginning to be ensheathed, and you can see that because of the green right at the root. Okay. Now when we look at the treated hyperglycemic fish, we notice a couple of things. The first is that the perineurium is not wrapping. If you can see the green cells, they're coming out and they're following along the notochord. This is the notochord here. But the actual wrapping isn't happening effectively. The other thing that you can notice, that Julia noticed, was that the axon itself looks very unhealthy. And so her second, after quantifying this and seeing that there was a significant difference in the motor nerves that had perineurium associated with it versus the motor nerves that did not, her second question was, well, what's happening at the level of the motor axon? If we're getting rid of this blood-nerve barrier, what's happening to the motor axon? And like you would expect, what we see, again, red is motor axons. This is a nonspecific neuronal marker that labels motor axons and the spinal cord. And what you see in the control fish is this very nice, healthy, tightly wrapped nerve, okay? But over here in the hyperglycemic fish, the nerve is actually coming apart. The motor axons are defasciculating. They are not healthy nerves. They're not going to be capable of sending signals to the muscles that need them. The other thing that Julia picked up on was that we have these, they're called neuromasts. They're a sensory organ in zebrafish. It's comparable to the vestibular system. And what she noticed was if you zoom in here and you see the healthy neuromast in the non-hyperglycemic, when you compare it to the hyperglycemic, it's very disorganized. And so this gives us an idea that there is a sensory component to this as well. So Amanda, she did some research with me this summer. She's been, like I said, she's been working in my lab for a few years now. She asked the question, well, what's happening on the sensory side? What's happening to the sensory neurons? So this is a different transgene. Again, the transgene is over here. This just tells us what components of the zebrafish are labeled. Here's our spinal cord. And in the control, you can see that the sensory neurons are located on either side of the spinal cord in what's called the dorsal root ganglia. And in the control, they're nicely clustered next to the spinal cord. But what Amanda saw was that these cells are actually migrating away from the dorsal root ganglia. So again, these are cells that are responsible for sending sensory information into the spinal cord. And then from there, that information either goes to the motor nerves or it goes up to the brain. So we have cells that are losing their way. They're migrating away from the spinal cord. They're not going to be able to do their job. When we look at the number of cells that are actually moving, we see that they're migrating away in droves. It's highly significant. So in terms of type 1, we have a good idea about the phenotype of what to expect. But type 2 is a little bit less clear because there was no model already established. And so when I was doing my postdoc, the last couple of months that I was there, I started just kind of messing around. I know that zebrafish glucoregulate in a way that is very similar to humans. And so I found some papers that showed different concentrations of glucose. And so what I did was I looked to see what these different concentrations of glucose that disrupt glucoregulation in zebrafish, what they would do to the peripheral nerve. And so this is a preliminary work that I did, but again, in green is the perineurial glia, and then in red is the motor axon. You can see in the control it's nicely ensheathed. In the hyperglycemic fish, it is very defasciculated. Likewise to what Amanda saw, when I look at the sensory neurons, they should be clustered around the spinal cord like you see in the control. But in the hyperglycemic fish, they're also migrating away. So let me just say how excited I was when I saw Julian and Amanda's research, because it was consistent with the small amount of research that I had done looking at the type 2 model, and that never happens. I've never seen data this consistent, which is really exciting. So we had to come up with a type 2 model. These are 3 of the students that worked on this component of the project, making this type 2 model. And so this is Derek Miller. He is now a medical student at Virginia Commonwealth University. Hannah Ennerfelt just started her Fulbright Fellowship in Sweden. She's studying motor neurons there. And then Rebecca Warfield, who is applying for a PhD in neuroscience as well. So what we did was we did a survival curve to see what the actual optimal amount of glucose that we could put these fish in to make them hyperglycemic without killing them. So we figured out that 60 was our sweet spot, 60 millimolar. And we found something really interesting, and this is going to be another area of research in my lab, and that is when we make these fish hyperglycemic, in terms of gene expression of insulin, the gene expression actually went down for insulin. So we may be causing some beta cell toxicity, and like I said, that's going to be an area of future interest. But ultimately, we made them hyperglycemic, okay? And so once we knew that we had a model that was making fish hyperglycemic, we asked the same sort of questions. The first being, what's happening to the perineurum? This is Sam Allen. She worked on this project 2 summers ago, and she showed that in hyperglycemic zebrafish, here's the perineurum again. In hyperglycemic zebrafish, we're losing the perineurum. So again, consistent between both models. Statistically significant, always a good thing. Hannah looked at the motor axons, and what she saw, this was really exciting, not surprisingly, you see the defasciculation like Julia saw, but also what she noticed was that we have cells, again, outside of the dorsal root ganglia. But she used a transgene over here that is a nonspecific label for all neuronal cells. So we weren't sure if these were actually motor neurons, which could be substantial, or if they were sensory neurons. And so we started with the low-hanging fruit and asked, are they sensory neurons? Because we know that in the type 1 model they are. And what we found was, when we used a different line that's specific to sensory neurons, we found that they are migrating out, the sensory neurons. But in other data that I collected, I also found that there are some motor neurons migrating out as well. And so we are messing with the boundary between the central nervous system and the periphery, which is really exciting. Jessie Todd quantified this data over the summer, so she repeated the experiments and counted the cells that were migrating away. But if you remember, I mentioned before that the neuromasts are populations of sensory neurons that are in the lateral line of the zebrafish. We weren't sure if these sensory neurons were actually migrating away from the dorsal root ganglia or from the neuromasts themselves. And so Jessie also spent time this summer counting the actual cells from the dorsal root ganglia to see if there was a change in the actual percentage of cells. And indeed, what she sees is that there was a decrease in the number of cells in the dorsal root ganglia. So we're pretty confident at this point that these migrating sensory neurons are actually coming from the dorsal root ganglia. And that really is an important point because we know how important these cells are for communicating information about the environment. So in terms of the conclusions from this, we've created a type 2 zebrafish larvae model of diabetes. And we've created a model of diabetic peripheral neuropathy for both the type 1 model and the type 2 model. And we see very clear phenotypes: axonal degeneration, perineural cell loss, and sensory neuronal migration. And all of this has set us up for what I really want to move into in the future, and that is looking at a small molecule drug library. So what we can do, we can exploit the zebrafish, we can take the eggs and grow them up in glucose or treat them, chemically ablate their pancreas cells. And then we can look for, let's say, axonal degeneration. We add these small molecules where the pathways are already known, and we can look to see if, say, for example, axonal degeneration is actually attenuated or if it's accentuated. And this might give us an idea about pathways that could be studied clinically. We have a number of other projects. I want to give credit where credit is due. Unfortunately, I don't have time to talk about all of these projects. Michael Kramer is going to be working with Stephen Habe and myself. He's going to be synthesizing a drug that we're going to use on the zebrafish. I'm really excited about that. We're looking at these models in adults. We're looking at them in old fish. So we're looking at an entire time span and seeing how the degenerative properties change across the course of the life. And then lastly, the two projects that I'm really excited about adding into these papers are the Schwann cell components. And so what's happening at the level of the Schwann cell, because that's something we haven't looked at in great detail yet. Oops. But I want to just briefly mention how phenomenal I think the students here are in terms of research. You know, I volunteered as a student researcher when I was an undergrad, and then, you know, did a substantial amount of research in the lab. But for us, undergraduate research was washing rat tanks, right, or washing rat cages and filling syringes. And here, our students have the opportunity to really get in there and manipulate things. And I mean, what an incredible opportunity. So for those of you that are interested in research, come talk to us, come talk to the faculty members. I'm going to shamelessly plug the Office of Undergraduate Research and Creative Activities. Come see us in the Academic Commons 233, because undergraduate research should be an integral part of your scientific career, or depending on what field you're in, it should be an important component of your academic career. So with that, I want to thank all of my current members, all of my past members. I want to thank my future lab members. My daughter comes in on the weekends and feeds with me when my students aren't around, my son. And I want to thank Salisbury University. Provost Olmsted has been a tireless component of the undergraduate research experience here. Dean Buss in the back, keep sending me those students, I'll keep taking them. And obviously Clifton's office, Henson, biology. I couldn't have imagined a more supportive environment when I took this job, and I'm so incredibly grateful for all the experiences that I've been able to have here. And I would be happy to answer any questions.
So you've clearly established a correlation between blood glucose levels and the sheet that protects the nerve. Do you have any idea about the molecular mechanism? So what is it at the cellular level that's being influenced by the glucose concentration?
So yeah, we're starting to broach that. We think that it's an advanced glycation end product buildup, either vascularly or intracellularly. And so that's one of the things that we are starting to look into. That's the goal. We just needed a phenotype to say, what are we looking at here? Because the molecular mechanism is obviously really the important underpinning.
I agree.
Yeah, obviously.
Any other questions?
Okay. Thank you very much. Thank you.
That concludes our presentations. Thank you for joining us.